Yeah. Better than getting COVID, though.
Yeah.
Sometimes I consider that, though.
No, don't consider that. Do not consider that.
Coming up, you start getting desperate.
What comes up?
That just delays the inevitable.
What comes up? What comes up now?
She was talking about what's more preferable, getting COVID or having to take finals. But I'm just saying, you still have to take finals.
Speaking of the finals, it's just the stress that they induce.
Yeah, I agree with
Like,
that.
I die from stress anyway, so.
Let's see.
Which one gets you first?
That, I get,
Yeah.
I agree. She's not wrong.
All right, let's see. I need my keys. That would be where our information is. Okay. Should just give it open. That board needs to be cleaned. All right. Oh, I need my chat.
Have you heard of the glycolysis rat?
I have not heard the glycolysis rat.
No.
No, you need to send it to me. Send it to me. My favorite thing like that is there's one. It's this Irish guy, and it's a Mother's Day song, and it's all about why you're slightly more genetically related to your mother than your father. But it's awesome. I'll have to like put it on our Moodle page or something. But it's like somebody sent it to me when I was in chemistry. I had just had Charles, you know, and it was like you're slightly more your mother than your father, and it's hilarious. It's hilarious.
I was looking to send it to you, but I saw this the other day, and I thought of you. I don't know if you can read it, but it's kind of small.
Let's see.
Is this like the picture? Because it's a cell wall.
It's a cell wall. That's good. That's good. That's very good.
It's so sad that I laugh at science puns.
No, it is not sad that you laugh at science puns.
It could be worse. I could laugh at math ones. Like,
That's just like my very smart people joke for science, like.
but it's sad.
It's C.
It's sad because they're funny to me, and then I'll tell them to someone else, and they just don't care. That's why they're sad.
That's true. When somebody doesn't care about a science joke, that is disappointing. Okay, let's see. I think that's all I have that I need. Okay, so I need to open up. Oh, I need to log into Clicker. I was like, there's something I haven't done yet.
So this is my freshman year of college, and when our professor showed us this, and it's been stuck in my head. It's actually very helpful for glycolysis.
Oh, awesome. Yes, do send it to me, please.
And Hannah's on there. She knows what I'm talking about.
Okay, let me minimize the chat. There goes attendance. Then I'll put the chat back up. Okay. All right, so I think we're ready to start. So I will start with your joke of the day. Are you ready for this?
Go for it.
So, you know, no matter how popular bacteria get, you know, they're just never going to go viral. That's it. That's it. Charles didn't like my joke this morning, so I was like, no, I'm not going to do that one. But they'll never go viral. I thought it was funny. The people in class are not impressed, FYI.
We're smiling behind
They're, oh, but they say they're smiling behind the
I
mask.
gave you
Okay.
bottom diss.
He did. Ross did give me a bum. He did. He did.
I'm
So I'm
smiling
happy.
approval, but you just couldn't see it.
Hard.
Okay.
Right, yeah, I know. These masks are really hard.
The masks are hard to tell when y'all are laughing or not. It is very hard. Okay. So we are starting chapter 9. The next exam is on chapters 9 and 10 only, right? So
I remember that. Then you said that we're just gonna add, we're just gonna tag on chapter 11.
we are tagging on chapter 11 to the final. That is correct. All right. So glycolysis is the name of the game. And we have to do a little bit of an overview of metabolism before we can actually jump into glycolysis. And then the rest of the chapter is really all about glycolysis. And so hopefully we'll be able to get through 9.1 and 2 today. It's really a big, broad overview of metabolism. And then if we're going to talk about glycolysis, what does glycolysis mean? What is
Breaking down of sugars.
Glycus means sugar, sweet. And then lysis means breakdown. So it's the breakdown of sugars. And so you have to understand the structure of sugars. And so I'm going to kind of ask y'all how much you know and how much you don't know about sugars. Okay.
I remember the basics, but remember the basics.
Well, we're going to review the basics. All right. So metabolism is our collection of all of our biochemical reactions, right? Everything that's going on. And we're going to use chemical energy to convert that into work. So we're going to have catabolic and anabolic. What is catabolic? Y'all can
Catabolic means
talk on the chat.
With oxygen.
No. Catabolic and anabolic. What's the difference?
Catabolic means
So catabolic is breakdown. Oh, why did that happen? Hold on. Catabolic is breakdown. Anabolic is the opposite, so synthesis, right? Creation, right? Build up, yes, okay. So catabolic pathways lead to the degradation, but how? And why? What's the point? What's the whole point of breaking down these molecules? It is to get energy. We need to transfer the energy that occurs inside of those molecules into something usable. And we're going to use NADH and FADH2 as carrier molecules, right? And then our anabolic pathways are going to use that energy that we eventually will get to the ATP. And we'll use it in the ATP form. We'll hydrolyze ATP. We'll harvest some of that energy. And we'll create reducing equivalents, right? So that we can synthesize all the building blocks that we desperately need for our cells to function all the time, right? So it's really important to remember that catabolic and anabolic pathways are happening simultaneously, always in the same cell at the same time. And so you have metabolites that occur in both the catabolic and the anabolic pathways that, that serve as substrates and products for different enzymes in those anabolic and catabolic pathways. So there's a lot of crosstalk.
So just a quick little recap. You're saying that since both reactions are taking place at the same time throughout in a system, so there are times whenever the signal from one gets perceived by the other. Is that what you're trying to say? Yes,
yes, and we're going to use some examples. The question is it that the catabolic pathways will have an impact on the anabolic pathways, and will the anabolic pathways have an impact on the catabolic pathways? And the answer is yes. There will be crosstalk that way. So when we talk about all of our pathways, we talk about a general term called flux. Flux is really the rate at which our substrates and our products are interconverted, right? And so we're gonna look at what controls that flux. And there are two primary mechanisms that control the flux. The activity level of an enzyme is one of them. Let me move this up because I want to write some stuff at the bottom. So tell me what can control, what can control the level of enzyme activity in a cell? This is one of the primary factors that's going to control our flux. So how do you regulate the level of enzyme activity? We talked about a lot of different ways, and so I want to list as many as we possibly can. It's a protein. Regulatory molecules is good, so that's catalytic activity, right? And catalytic activity is then regulated by regulatory molecules. That's true, right? Also, how is catalytic activity regulated? There's another way too. Like you could bind a regulatory molecule, or something could do something to you, to that enzyme. Change it covalently, covalent modifications. Yes, add a phosphate group, something like that. Remove a phosphate group. Okay.
Add or remove a phosphate, or just remove?
Add or remove, and it doesn't have to be a phosphate group. But that's kind of the examples we've been talking about so far. But there are other examples. So catalytic activity is one way. What's another way? Proteolytic processing. Yes. So we'll say proteolytic processing. Yes. So pH and temperature. So we can say environmental conditions. And this is also, what else is included in like pH, temperature? That's true. pH, temperature. What else? Environmentally.
Pressure. Pressure.
No, your pressure is really not going to change. That's kind of a constant. But think about concentration has to do with more like protein synthesis. Protein synthesis. That will control concentration of the enzyme. That's true. But think about environmental conditions, right? If the enzyme is not in the same place as the substrate, is it going to happen? Is our reaction going to happen? No. So really cellular location, right? Location. It's like real estate.
I know location's a big deal because that's what they all do. These home improvement shows my mom watches.
Same thing in the cell. Okay. So and then the last one, if we talk about protein synthesis, what's the opposite of protein synthesis that we also have to worry about? What? Yes, degradation, turnover. R-E-D-A-T-I-O, something like that. I-O-N. It's like sometimes the pen doesn't pick up what I'm writing. Okay, so those are all good examples of how we can change the level of the enzyme inside of the cell. So that's great. That's the enzyme. All the things we just described are the enzyme. Now let's talk about the substrate. When we talk about metabolism, where are our substrates coming from? There are only two places that our substrates can come from. Any ideas? Y'all did read the section, right?
Yes. Sugar.
Where do you get sugars?
Oh, like
Where do you get sugars?
food.
From food, from our diet, right? That's true. You don't make sugars, do you? Are you a plant? It would be really cool. I'd love to photosynthesize. I'd be green though. But not necessarily, but you know.
I don't know. That sounds,
be cool. All right, so where else can you get it?
You get your diet, you can get it from.
If you don't eat for a while, what is your body going to do? You're going to go into starvation, and so you're going to start what? You're going to get it from your liver. What do you get from your liver, Hannah? Glucose. So you can go to your storage. You can go to your storage areas. Like glycogen is inside of the liver that gets converted to glucose. That's right. Good. Good. So like energy storage. E-N-E-R. Energy storage. right? So either you get it from your diet or your body has stored it and then you're going to convert it, right? So let's talk about those two things, right? Okay, we have two little pathways here. Can I completely minimize that? No, it has to sit here. Ah. Okay, so I have two pathways, A and B. Take a look and tell me what's going on that's different. The big arrow means that this pathway is active, and the little arrow means that the pathway is basically suppressed. So if I have a suppressed influx here of glucose, what's going to happen? That means that I'm fasting. So maybe this is overnight. I wake up in the morning and I haven't eaten breakfast yet. My blood sugar levels are low. So my influx is low. That's what the little arrow means, right? What's going to happen? Am I going to be synthesizing glycogen? Do I want to go and store glycogen? No, because I don't have sugar in my bloodstream. So at this point, in order to maintain a sufficient blood glucose level, I'm going to go through glycogen degradation from the liver. Right? Now, I convert that glycogen to glucose and I can pump it out into the blood to meet my body's energy needs. At the same time that this is happening, your body can also take pyruvate and go through gluconeogenesis and make glucose. So if you look here, what kind of a pathway is this? Anabolic or catabolic?
Degradation.
Right here, glycogen to glucose.
That's anabolic. Catabolic.
Catabolic, right? Catabolic. Okay, so we're going to promote a catabolic activity. How about here? We're going from pyruvate to glucose. Catabolic or anabolic? Anabolic, we're making. So do you have catabolic and anabolic processes happening simultaneously inside of a cell? Absolutely. Okay, now you're going to break your fast and you're going to have breakfast. Hence the name, breakfast. Right? So you eat breakfast. You eat breakfast. What's going to happen? Oh, I got an influx of glucose. So if I bring in a lot of glucose, then I'm going to store some of it, right? I'm going to store it where? In the liver as glycogen. I'm in the process of storing it. Do I want to break it down?
Nope.
Nope. So they're going to flip-flop. Well, okay, so there's a limit. There's a limit because if you eat more than what your body can store in the liver, then you're going to start storing it as fat. So you're always going to store it, but it's how you store it. So it'll go to the liver first. So then we have glucose, and glucose, right? We're going to break that down in glycolysis, and we're going to make pyruvate, and this is how we get energy. So at the same time, do you want to be making glucose with gluconeogenesis? No, you absolutely don't. So what you want to do is use the energy that you have from your glucose from your breakfast, and then anything that's left over, you're going to store it for later use. So you have anabolic and catabolic going on simultaneously. Okay. All right, now, if you look at all of the metabolic pathways, this is what they look like. Right. This does not look like something you want to study, right? But I show you this not to scare you and not to tell you you're going to memorize all of it, because you're not. But it's important. We're going to take one pathway at a time when we study. But it's important to know that this pathway is like one tree in the forest, right? And if you want to understand how the forest ecosystem works, yes, you need to understand how each individual tree works, but then you also have to know how the entire forest interacts together to make that one ecosystem. Okay, so we're always going to look at one pathway and then see how it fits into the big picture. look at another pathway, see how it fits into the big picture. Okay? So it's like the forest through the trees, right? Okay, but they're very, very independent. So do you see how it's not just like one pathway by itself and another pathway and another pathway and another pathway? Do you see how interconnected it looks? So you have to understand them all to understand the big picture, right? So the two ways that we control this crazy complex mess is by enzyme activity levels and substrate bioavailability. And we talked about the different ways that we can do that. Now, I'm going to show you a simplified version of this that I think you're going to like better. All right. So here's our simplified version.
Still a lot.
Yeah, well, we're going to take still one at a time. We're going to still take one at a time. So what you can see here are, these are our four primary biomolecules, right? Proteins, nucleic acids, carbs, and lipids. So what you're going to notice is that we have lots of things that are shared between the different pathways, right? We have metabolites that are shared between the pathways. That's our blue ones, right? We have small biomolecules down here that are shared between the different pathways, like NADH, FADH2, O2, CO2, right? So you're going to see these molecules over and over and over again. And we're only going to take kind of one at a time. All right. So don't get overwhelmed, but it's important to kind of keep a good big picture. And this is a good big picture for you to kind of keep in your mind. So we can take this map and we can divide this map into two major divisions. So the first division is the red division. So the red division here, right? This is our energy conversion, right? So glycolysis, citric acid cycle, oxidative phosphorylation, photosynthesis, and carbon fixation. These are all energy conversion. So we're going to oxidize some simple sugars through a series of redox reactions, right? And it's going to culminate in ATP synthesis. That's our goal, ATP synthesis. Now, we also have these blue pathways. So what are these blue pathways? These are the ones that control the synthesis and degradation of macromolecules. So those are our two major classification groups. And so we're really going to focus on energy conversion for the rest of the semester. We're going to focus on energy conversion. And then we're going to talk about in Biochem 2, our synthesis and degradation pathways, which are really, really cool. What? Yeah, the ones in blue. And they'll come back and it'll keep pulling from glycolysis. It'll be dependent on your understanding of these will be dependent upon your understanding of these.
So blue is for macro.
Blue is synthesis and degradation of macromolecules. Red is for energy conversion.
Oh, okay.
Okay.
So the image on the left is basically it's just used to help like paint a picture for the chart on the right.
Yes. Yes. Okay. So when we look at our pathways, it's really, really, really important that you remember chapter 2, right? Chapter 2, you had to do all of that. We talked about energy, right? And coupled reactions and delta G. And you remember all that kind of stuff? Hopefully. Yes. Okay. So when we look at the energy of a reaction, what we're looking at is if it's free energy, is it spontaneous or is it non-spontaneous, right? And you remember our Ratlink equation. Did I call it a Ratlink equation?
Yeah.
Yeah? You don't remember. This is just what I call this equation so that you can kind of remember everything that's in it. RT ln of K, rat, link.
Was it like, is it positive or negative means it's spontaneous?
Negative is spontaneous. Negative is spontaneous. So if you remember delta G, what is this little knot and what does this little dash mean? prime and the not. So what is delta G not? What are those conditions? Anybody? Well, no, like the conditions inside of a reaction that are, these are our equilibrium, yes, but oh, here we go, 1 atm, right? 25 degrees Celsius. What else? One more, let me see if they wrote it, no, one more. Concentration of everything is 1 molar. Is that what happens in the cell? Yeah, no. Means, okay, so now we need to talk about the next one, which is delta G prime. So what do we talk about when we're in physiological conditions? That's right. pH is 7. We have a concentration of water. Remember that? 55.5. If you have magnesium present, you have a magnesium 2+ concentration, right, of 1 millimolar. Remember this? But is every reaction those conditions?
No.
Nope, absolutely not. So you can have something that is unfavorable, but how the reaction is completed in the cell will change its delta G.
What's the thing for the
Mg2+ is 1 millimolar. So if the reaction's unfavorable, a lot of times what the cell is going to do is couple it, right? We've talked about that already. So what's the definition of something that's coupled? We're going to use shared intermediates. So here's an example from glycolysis. Phosphoenol pyruvate plus water goes to pyruvate. This is a favorable reaction, but we want to be able to generate ATP. Usually we go in the opposite direction because look at this, it costs 30 kilojoules per mole to make 1 molecule of ATP. That's pretty expensive, right? So what are we going to do? We're going to couple it to a reaction that's spontaneous. And so that coupling leads to our overall negative delta G so that we can make this reaction happen. So we're going to talk about exactly how that works on the next 2 slides. But the other way that the cell will make an unfavorable reaction happen, and the reason why coupled reactions work, is because a lot of times in metabolism, the metabolic enzymes are linked together physically. They're physically next door neighbors. And so if you convert your substrate into one intermediate, it doesn't go anywhere. It just goes to the next enzyme and then goes to the next enzyme and goes to the next enzyme. And so you end up with this no loss of substrate concentration as you're making all these different intermediates, right? And so it helps your coupled reactions actually go. All right, so if we look, right, at what actually happens in the cell, right? This is our delta G for our normal cell conditions, right? That's a combination of what happens under physiological conditions plus RT-ln of the concentration of the actual reactants and substrates. Because are our concentrations usually at equilibrium in a living system? No. What happens when they reach equilibrium? You're dead. Yep, exactly. So we don't really have a K here. What we call when they're at actual concentrations is Q. Okay, so Q is the wannabe K. That's what I call it, right? It's not at equilibrium. And so the concentration of our products and our substrates is going to have a massive impact on whether or not our reaction is actually favorable inside the cell or not. Massive. So if we look at one example, we're going from glucose 6-phosphate to fructose 6-phosphate. This has in standard conditions, standard physiological conditions, a positive 1.7 kilojoules per mole. Spontaneous or not? Not spontaneous, right? But if we look at the concentrations of the two, right? Here's our substrate and here's our product. What do you notice about the concentration of the two? Well, not the same. I mean, look at your exponents the same, but tell me what's the difference between the two? What's 8.3 divided by 1.4? Somebody. I don't have a calculator. Anybody. A phone. 5.92. So almost 6 times less of our product than our substrate, right? So if you plug those values into Q and you solve for the delta G that actually occurs inside of the cell, what you're going to get now is a negative delta G value. Why? So you know about, right, Le Chatelier's principle, right? So if we have substrate concentrations that are so much greater than our product concentrations, right? Massively higher. And this is actual, not equilibrium. This is actual. What's going to happen to the value of Q? Is Q going to be greater than 1 or less than 1? Q ends up being less than 1. That is going to have an effect on your overall delta G, and it's going to make a negative delta G. Right? Because you're taking the natural log. So it's going to give you a negative value here. Does that make sense mathematically? Right? Okay. So what drives this reaction forward is that our product here is a substrate for the next reaction. So is our product going to hang out once it's made? No, it's got somewhere to be. It's being used up by the next reaction. So the conditions that drive our reaction forward are going to be having those shared intermediates. If the intermediates weren't shared, this wouldn't go. It's because you're using up the product, and so you're making the reaction shift towards the product. So the question is one more time. Okay, so why is it important that reactions are coupled? Right? This is our big thing. We know coupling reactions is important. The question is, can we prove why? And the answer to why is because it makes our product concentrations drop really low. Right? You're still eating your lunch. You're still getting substrate to come in. But as soon as you start metabolizing that substrate, right, it doesn't stop at one reaction. It goes on to the next. The product from one reaction becomes the substrate of the next reaction. And that's the definition of an intermediate. So if we use up our products, then we're going to have a shift of the equation towards the product in. And why do we have that shift? Because of Q. And we can prove that mathematically by showing that we get a negative delta G value, an actual delta G value, because of this difference in concentration. Yeah? Okay. So now we kind of have an overview of metabolism. Y'all ready to start talking some sugars? Let's do it. All right, so this is where you're going to kind of have to tell me how much you know and how much you don't know, because I kind of want to know where we're starting from. So a monosaccharide means one sugar unit, right? And usually you either see them, have you seen them in a Fisher diagram? Do you know what a Fisher diagram is? Yeah, and the chiral carbons are the ones that are at the T's. y'all remember this? Yes? Some people in class are saying yes. I've got no response on the chat.
Sorry, can you repeat that?
Have you seen a Fisher diagram of sugars and seen how they convert
If I have, I probably forgotten what it was.
briefly?
I've forgotten what the name is. I don't know.
Sort of, kind of. Okay, so I have a good review for us, so I think it's a good thing I planned a little review. All right. So glucose is an example of a monosaccharide, right? This is a monosaccharide that is a polyhydroxyaldehyde. What does that mean? We have many hydroxyls and an aldehyde, right? It has a lot of hydroxyls and it's an aldehyde. Do you remember what an aldehyde is? So you have a carbonyl carbon, right? And then what's bound to the carbonyl carbon? We have an R group and a hydrogen, right? Okay, that's an aldehyde. Now, fructose is a poly. Why are there multiple D's in there? That is not supposed to be multiple D's in there. Sorry about that. It's a polyhydroxy ketone, all right? So pardon my spelling. So if it's a ketone, what's a ketone? We have another R, right? R1, R2, and we have a double bond oxygen, right? So you have two R groups. Y'all remember that? Yes. Woo. Okay. So when we eat these things, right? They taste sweet. Why do they taste sweet? You could have said that before this class, but if you remember the last chapter, right? What did we say in the last chapter? How do we get something to taste sweet? We have a ligand that activates a receptor. What receptor does it activate? So the sugar is our ligand. Yes, William, ligand. And our ligand activates a GPCR, right? Okay, so does every sugar activate my receptor in the same way? No. If you look, different sugars have different levels of sweetness, right? Have you ever noticed on lots of food containers, it'll say made with high fructose corn syrup?
All I know is
Every food container has that. Why? Why does every food container have that? No, they're business people. It's business. So let me explain the business to you. All right. Glucose and fructose are both sugars. They both have the same C6H12O6. They're both like that. One's a ketone, one's an aldehyde, right? They bind to the receptor with different affinities. So fructose binds with a higher affinity to the receptor than glucose. So guess what? The same amount of fructose tastes sweeter. More than 2 times as sweet.
so basically,
Binds more. Fructose has a higher affinity.
fructose has a
Higher affinity. And so it's actually cheaper because you have to put less sugar, right? We're not defining what sugar it is. We're just saying you have to put less sugar in to get the same level of sweetness. So they can spend less money and put fructose instead of glucose, or a combination of the, usually it's a combination of the two, and save money in manufacturing whatever food product it is. Is what? No, they're all. The definition of natural is
pretty loose ended.
very loose ended, right? Very loose ended. All right.
At the end of the day,
Why not use glucose? I mean, if you only need less glucose to make something.
No, you need less fructose. Fructose binds more tightly. So fructose is going to taste sweeter than glucose. So to get the same taste, you have to use less fructose than glucose. So if they just put glucose in their food, they would spend more buying the sugar to make their food than if they buy glucose and, buy fructose and put it into their food. Yeah, kind of crazy. All right, so here are our two structures for glucose and fructose. Remember we said glucose has an aldehyde group, right? So here's your carbonyl carbon bound to the oxygen. So it has a hydrogen, and then this is the rest of the R group, right? So whenever you're numbering your sugars, you start at the carbonyl carbon. If we look at fructose, fructose has our ketone. So remember, this is how I remember a ketone versus an aldehyde. An aldehyde is all alone. There's only one R group, but a ketone, like a lock and key, you need two things. So you need 1, 2 R groups. That's how I remember. So here, if you look at your ketone, you'll see that there are carbons on either side. So you're going to start numbering on the carbon side that has fewer carbons, the R group that has fewer carbons. So here we're going to number this one 1, and then our carbon bound to the carbonyl, our carbonyl carbon, is carbon number 2, okay, and all the way down. So these things are going to form ring structures, and we're going to talk about how they form ring structures in just a minute. But for now, I really want to focus on our Fisher diagrams here. So it's really in the cell and in solution in equilibrium between the straight chain and the cyclic form. So if I were going to classify glucose as a type of sugar, what type of sugar is glucose? It has an aldehyde, so it's an aldose, but how many carbons does it have? 6, so we really call it an aldohexose, O-S-O-S-E, not S-is, aldohexose, right? The O-S-E means sugar. So how should we name fructose? What classification? Look at this one. Should be a keto. How many carbons? 6, so hex, and then it's a sugar, ose. Okay. So all of our sugars, no matter if they're aldoses or ketoses, they're all going to have this basic structure, and they're all going to have one carbon that is not going to participate in the ring. So in order to make the ring, you're going to have a reaction between this carbonyl carbon and the oxygen in the hydroxyl that is in the chiral carbon, that is bound to the chiral carbon that is farthest away from the carbonyl group. So it's going to be this oxygen right here. It's going to be this oxygen right here, which means that this CH2OH group is going to sit outside the ring. So see how number 6, number 6, number 6, number 6. In the case of a ketose, we're also going to have another one that sits on the outside of the ring, and that's going to be carbon number 1. So carbon number 1 is going to be here. Here's the carbon that was bound to the carbonyl, right? Well, it was the carbonyl carbon. Now they call it the anomeric carbon, right? That's carbon number 2. Are these terms familiar to y'all?
Somewhat. Somewhat.
Yeah, organic. You should have gotten this inorganic, right? All right. So what do all of these? Why are these carbons not listed, but this one is? Why do we just list them as like a plus sign? Why don't we put a C?
Because
They have, not only do they have 4 bonds, what's important about those different bonds? They're all different. I gave it away. They're all different. So they're chiral carbons. So how many chiral carbons are in glucose? 1, 2, 3, 4. How many chiral carbons are in fructose? 1, 2, 3. Okay. It's important that you know these terms.
so you're saying they're shown on the graph because they're shown on the structure because they're chiral carbon.
They're shown as plus signs. They're shown as straight lines intersecting because they are chiral carbons. It's an easy way to look at the structure and know that that's a chiral carbon.
So what about the ones that aren't?
They are not chiral. They have 4 bonds, but look, this is bonded to 2 hydrogens, so it's not chiral. Same thing, bonded to 2 hydrogens, not chiral. Bound to 2 hydrogens, not chiral. Chiral means you have 4 different things attached. Okay, so now when we look at these Fisher projections, right, we can label these as either the L or D orientation. Have y'all heard of this before? We're like squinting eyes in here, like maybe.
Do you remember?
I don't remember it. Okay, okay, not so much. All right. So the L and D designation are all about the location of our chiral carbon, right? And where the hydroxyl is attached to this, this farthest carbon, we call it the penultimate carbon. So this is a chiral carbon that is farthest away from our carbonyl carbon. So penultimate is farthest away from carbonyl carbon. So we look at that carbon and we say, okay, we know we're going to have a hydroxyl attached to it, but is it on the left or is it on the right? If it's on the left, we call it the L designation, right? L and L. L is left. Right, we're going to call it D. Why we couldn't have called it R? I don't, well, you know, whatever.
So like, depending on the location, so the chiral carbon. So like, if the
I'm pretty
carbon-
sure we skipped this chapter because we focused on carbs. We skipped about I tutored it. Okay. Okay, so that's good. That's good that we're doing this review
How do
then.
you know where? How do you know if the carbon is pointed left or right?
You don't know that the carbon is pointed left or right. You know that the hydroxyl is either on the right or the left. So you could have a, let me draw it. I wish I could scooch over that way, but I can't, right? So here's our carbon. If I want to draw the L, so if I want L glucose, right? So what I'm going to do is I'm going to have a hydroxyl on this side and a hydrogen on this side. And so I'm going to basically flip-flop everything. So this is going to be an OH. This is going to be an H. This is going to be an OH. This is going to be an H. This is going to be an OH.
Oh, wait, that
This
one
is going to be an H.
That one's just, oh, wait.
Oh, sorry. No.
Number 3 has a.
Number 3 is just an H. Sorry. Thank you. OH. I'm just going to town. All right. CH2OH. So do you see how here we're on the right, here we're on the left? Does the position of carbon 3,
Like the hydroxy is on the right, all the other hydroxys are on the left.
that will tell you if it's a different sugar. It'll tell you it'll be a different sugar. It'll be an enantiomer. I'm going to show you that, and it'll be called a different sugar. But whenever you do this, when you're going between D and L, so when you switch between D and L form of the same thing, so this is glucose, right? These are called enantiomers. E-N. If I could spell, that would be wonderful. Enantiomers. They're the same sugar. They're mirror images of one another. They're non-superimposable mirror images.
So like sugar, they're mirroring to each other.
They are non-superimposable mirror images. Hannah, did that answer your question? I typed what you said, okay. Right, so these are both glucose. This one is D-glucose. You can't even read that. This is supposed to say glucose. I was too jammed up, right? So this is D-glucose, this is L-glucose. It's still the same sugar, but they're in different orientations. Now, I'll tell you that your body is only going to work with D-glucose. And almost every sugar out there that is natural is in the D form.
Happens if we get L sugar.
Your enzymes cannot metabolize it, and you would die. If that was the only food source you had, you would die.
So basically, we can only digest and metabolize the glucose.
Correct. Your enzymes are specifically designed. How precise is the active site of an enzyme? Very, very precise. If you had a mirror image of the molecule, it won't catalyze the reaction. And so it's very important that everything is in D. Otherwise, you would have to have two sets of every single enzyme inside of your body, one that would handle D and one that would handle L. Your body can't do that. It's super cool. Okay. So, right? You can have monosaccharides that differ only in the hydroxyl position around one of the chiral carbons. This is what you were asking about just a second ago. But we're going to call it something different. So if you started out with D-glucose and all you did was swap the hydrogen and the hydroxyl on carbon 2, right? That's carbon 1. This is carbon 2. What you're going to see is that we're going to call it a different sugar, and we call these two sugars epimers of one another.
So only one.
Only one.
Only one chiral can have an L structure. Only one chiral carbon can have an L structure.
Only one chiral carbon. Every single sugar molecule has an L structure and a D structure. If you select out the D structure and you take one of those chiral carbons and you flip-flop the hydroxyl and the hydrogen, you've made a new sugar, and that new sugar is an epimer of the original D sugar. It's still going to be D unless you swap this one, right? So here's another. This is ribose. Ribose, if we swap carbon 1, 2, 3, if we swap what's around carbon 3, what we're going to get is xylose. So these two are epimers of each other, and they are called a different sugar. The last one. The last one, the penultimate. It's not really the last one. It's not really the last one, because this is really the last one. It's really the penultimate, which is the chiral carbon farthest from the anomeric carbon. Okay. So now let's talk about how those simple sugars turn into, so how is D mannose D? What are you doing? It's D, it's still on this side. This determines if it's left or right. If it's on the right side, it's D. Here's right, here's right, here's right. They're all D. Yeah, you got to look at, this is the penultimate. This is the penultimate carbon. If you want to know if it's D or L, you need to look at the penultimate carbon to determine if the hydroxyl is on the left or the right. If it's on the left, it's the L. If it's on the right, it's the R. And it depends on what sugar it is, which carbon it is, right? So that's why we just call it the penultimate. That's why we have a different name for it. Okay, so our simple sugars can undergo an intermolecular reaction, which means it's going to react with itself to form either a pyranose or a fructose ring, okay? When it turns into a ring, it's going to give stability to the molecule. So typically in the cell, what you're going to see is most of the glucose is going to be in the pyranose form. So what happens is this hydrogen right here, I wish I could zoom in. I don't know how to zoom more. How do I zoom more? What little thing? Wait, I think I could do it. Can I do it here? No. What's the little thing though? This? I don't know. See, I need to figure this out. Because the pen is so big, the pen width is so big. Okay, so you have your hydroxyl and your penultimate carbon. That is going to form a bond with the carbon in the carbonyl carbon, and with the carbonyl carbon, right? So in this case, it's carbon 1 and carbon, that's 1, 2, 3, 4, 5, right? So if you're going to make a ring, what things are going to be in the ring? Well, here's your carbon 1, your carbon 2, your carbon 3, your carbon 4, your carbon 5. Now, carbon 6 is not going to participate in the ring because the next thing in the ring is going to be an oxygen, right? So you're going to have a 6-membered ring, but one of the members is going to be an oxygen. That 6 carbon is going to stick out. So what's going to happen is this oxygen right here is the red oxygen, is this one right here. I just like wrote over it, but it's this one. So this oxygen is going to form a new covalent bond to this anomeric carbon. So the carbon that was in the carbonyl is going to now be called the anomeric carbon. So it's got a new name because it's no longer a carbonyl carbon. So this anomeric carbon now has how many bonds attached to it? A hydrogen. Now I'm bonded to the oxygen. Here I'm bonded to a carbon, and then I have a double bond to an oxygen. How many bonds is that? Too many, right? No, 1, 2, 3, 4, 5. That's 5 bonds.
So they're all 18 bonds.
So what's going to happen is that you're going to lose one of these bonds, and so you're going to have a single bond to the oxygen, and then this hydrogen is going to hang out now with this oxygen. So what you end up with is an oxygen in the ring that has 2 bonds, 1 to each carbon, and then this anomeric carbon, which was the carbonyl carbon, is now bonded to an oxygen, and that oxygen picked up the hydrogen that was over here on this hydroxyl. Now, when this happens, there are 2 ways that this can happen. When you look at a ring's 3-dimensional. And so once you form the ring, you can't break the ring. We always say that the top side of the ring is the side that has this extra carbon. So if the hydroxyl group adds opposite, so if it's pointing down, what it's going to be is the alpha configuration. So it's trans. Y'all remember cis and trans, right? So trans is now going to be called alpha in a ring. If the hydroxyl binds and it is cis, it's going to be beta. So here, my hydroxyl that's on the anomeric carbon is on the same side of the ring. Not necessarily. Not necessarily. Hmm? It can't. It can't. That's why it's going to lose one of these double bonds to the oxygen. That's why. Yes. Okay, so I will stop right there. We're like almost done with sugars. So we'll finish sugars on Monday. Which OH in the ring is trans to the carbon? Okay, so we're always going to compare this one to this one. So this one to this one. So it's the one bound to the anomeric carbon that we're going to compare. So they're depending on the conditions and all the things that is going on, you do have some stability with the beta form, but it kind of depends. So I don't want to say it's always going to prefer the beta. It doesn't matter. You don't have to know that. Okay. All right. If anybody has questions on this, I will answer it, but you are free to go in 2 weeks. In 2 weeks. Y'all have a good weekend too. Anybody have questions? I'm happy to answer. We got something in class.
I have a question, Dr. D.
Oh, wait, we're gonna. All right, so whoever's on Zoom, go ahead.
Okay, so I think my Zoom cut out for a second. So if the penultimate carbon changes to just that one and not the rest of the chiral carbons, that's still not considered L, right? Or D because it's not a mirror image.
So you're talking about, let me go back. You're talking about here, right? We're not looking
Yes.
at the other hydroxyls. We're only looking at the penultimate hydroxyl.
Yes.
Correct.
So like if that 5th carbon changes, but none of the other ones do.
Wait, say that again?
So like where your mouse pointer is right now, that
Huh?
5th carbon, if that one changes to the other side, the hydroxyl group changes to the other side, but none of the other ones change,
Yes.
that doesn't switch its conformation from D to L, right?
Right. So if I wanted to draw this molecule in the L configuration, what I would draw is this. So I have 1, 2, let me extend the page. I can't. 1, 2. What am I doing? 1, 2, 3, 4. I have 4. Oh, this is glucose. I'm like, I don't even know what I'm drawing. This is glucose. Okay, so now I want to switch everything. Everything's going to be a mirror image. So anything that was on the left is now going to be on the right. So this is going to be a hydroxyl. This is going to be a hydrogen. This is going to be a hydroxyl. And this is going to be a hydroxyl. So now, if I look at my penultimate carbon, and I look at the side that my hydroxyl is on, it's on the left. So this would equal to L glucose.
Okay, yeah, that's what I was asking. So like it has to be a complete mirror image.
It has to be a complete mirror image. Otherwise, it's a different molecule.
Would it just be an epimer then if just the penultimate carbon changes and nothing else?
It would be an epimer. There are different types of epimers. There are epimers that are only different at one chiral carbon, and then there are epimers that are different at multiple chiral carbons. And then if you have all the chiral carbons that are epimers, then that is an enantiomer.
Okay.
So L and D.
That's what okay, yeah, that's what I was asking. So thank you.
Sure, of course. Anybody else? Yeah, we're going to finish 9.2 on Monday.
I have a question, Dr. B.
Sure.
Okay, so whenever you're referring to the like L and the D conformations, does it matter in terms of like the ones with the ketones? Does it matter which way you direct it, or is that only whenever you're reflecting the mirror image of it?
You don't have to worry about, like, it would be correct either way. You can have these things can move around, but the things that are on the chiral carbons cannot. So you could actually draw this and it's still the L form.
Okay. So like, not that it matters, but wouldn't the ketone typically face away from the hydroxyl group if you were being picky about it, or?
It's not, neither one is shown in 3 dimensions. Neither one is shown in 3
I got
dimensions.
you. I was just asking. I know it's a little bit much, but.
No, that's okay. That's okay. So if you were going to model it in 3 dimensions, absolutely, it would want to be farther away from the hydroxyl. But these structures don't show that. So you could technically draw it either way because they can move. The chirals can't. But the carbonyl and the carbon, the bonds around it can move around.
Okay, thank you so much. Have a good weekend.
You too. Anybody else?
Dr. B.
Yeah.
What's keeping those chiral carbon sigma bonds from rotating to different formations? Is it steric hindrance or something?
It's not steric hindrance. It's how the molecule was formed. Like you can't, if you were to build this, like this is hard because it's COVID and y'all can't actually touch a model. But if you, like, they have some things online that are like 3 dimensions that you can manipulate and you can turn and see. If you build it and then rotate it in 3 dimensions, what you're going to be able to see is that you can't rotate the molecule to swap the hydroxyls and the hydrogens. Like you physically can't do it. It's a trigonal conformation on that chiral carbon, so you can't. Does that make sense?
Yeah. I think I was just thinking about it in 2D for a minute, but I
Yeah.
remember.
It's hard that way. It's really hard to understand when you can't see it in 3 dimensions. And unfortunately, I can't give y'all things for everybody to touch.
I also have an unrelated question if you have a minute.
I have a minute, but somebody else in the classroom has a question too.
Yeah.
Is it a quick one?
I was just wondering if we knew what dictates the limits of like when someone runs out of being able to store glycogen and starts storing fat.
There is a limit. There is a limit, and it's slightly variable from person to person. But there's an absolute limit to what your liver can do.
Mm-hmm.
You're also going to store some glycogen in your muscles. So it depends. So it also depends on your muscle mass.
So it depends on how big you are, basically. Is that it?
Yes. Yes.
So you can't, if 2 people are the same size, could you have like one with more enzymes, converting enzymes to get more glycogen or something? Or is it solely on size?
I mean, you're going to have a little variation, but not a drastic variation. No.
Okay, that was my question.
Yeah. Okay. Anybody else on Zoom?
Yeah, I have a question about lab later today.
What you got?
I'm trying to, well, the one we're turning in today.
Yeah.
Moodle, do we turn in under lab number 4 lipid purification or lab number 5 enzyme purification?
Lipid.
Okay.
Lipid. But you could turn it in class if you want. It doesn't matter.
Okay. Thank you.
All right. No problem. Anybody else? Okay. So I'm going to go ahead and end the Zoom. Y'all have a great weekend. Oh.